I said everything gonna be alright. I said everything will be alright.
Everything's gonna be alright. Good day wherever you're listening from, and welcome to Indoor Air Quality Radio. It's IAQ Radio for Friday, August 5th. 2016. We're back live this week. It's episode 425. My name is Radio Joe Hughes, and I'm coming to you live from Central City, Pennsylvania, Studio D. Our engineer, John, you gotta have faith, is back at the controls, and joining me from Studio C back in McKees Rocks is the Z-Man, Cliff Zlotnick.
Hey, Joe. Hello, everybody.
Good day, Cliff. Just back from summer camp. Had a great time at the Building Science Corporation's 20th annual summer camp, and actually got to see today's guest do a presentation at summer camp. We're going to talk a little bit about that and about a bunch of other issues today with Dr. Mark Hernandez out of the University of Colorado. Before we get started, though, we've got to thank our marquee sponsors.
John Don Products, where restoration and abatement contractors shop. Visit them at their website, jondon.com. That's jondon.com.
Clean Facts, the #1 information source for cleaning and restoration professionals. Check them out at cleanfactswithanx.com.
IAQ.net and Healthy Indoors Magazine, a free online digital magazine for industry professionals and consumers. Subscriptions are available at IAQ.net. Please be sure to thank our sponsors for their support of IAQ Radio when you inquire about their services and products.
And last but not least, please visit the IAQ Training Institute website for the most current dates for the training you trust at IAQtraining.com, and check out our fall event. The Healthy Buildings Summit 3 will be October 20 and 21 in Seven Springs, Pennsylvania. All right, Cliff, let's turn it over to the Z-Man for today's IAQ Radio trivia question.
Thanks, Joe. Win a cool prize by outcompeting fellow IAQ Radio listeners and being the first person to correctly answer the IAQ Radio trivia question each week. Submitting your answer is easy. Either email it to czlotnick@cs.com. If you're listening to the show live, you can also text in the answer via your computer. Congratulations! To both John Lapotaire, Indoor Air Quality Solutions, Orlando, Florida, and Victor Cafaro, Richmond, Virginia, who tied on answering our last IQ Radio trivia question. The IQ Radio trivia question for Friday, August 5th, 2016, has been sponsored by Ideas, the solution chemistry company creating unique solutions to odor removal, surface cleaning, and decontamination problems. Now for this week's IQ Radio trivia question. Name the word defined as the study of airborne microorganisms, pollen, spores, and seeds, especially as agents of infection. Back to you, Joe.
Okay, thank you, Cliff. Dr. Mark Hernandez received all his degrees and did a postdoctoral tenure in civil and environmental engineering department at the University of California at Berkeley. After several years of civil engineering practice, he joined the University of Colorado faculty in 1996, where he is now a full professor. Dr. Hernandez is a registered professional engineer and an expert on the quantitation and remediation of bioaerosols. A generation of his research lies in characterizing the biological aspects of air pollution, both indoors and out. With respect to environmental investigations, his aerobiology characterization work has focused on large-scale disasters, including bioaerosols generated by major metropolitan floods. Dr. Hernandez's research group is based in an environmental microbiology laboratory, which also houses the largest bioaerosol chamber in the United States with full environmental controls. Since its commissioning, this laboratory has been active in aerobiology research supported by the National Science Foundation, the National Institutes of Health, the US EPA, and various private and public companies. Dr. Hernandez was recent recipient of the Lindbergh Foundation Environmental Award and a National Science Foundation's Early Career Award for bioaerosol research. We are pleased to have him with us on the show today. We've got some intro music for Dr. Hernandez.
Growing mold in my heart, growing mold in my brain, growing mold all around me and feeding me away.
All right. Hello, Mark. Do we have you on the line?
You do.
All right. Great to
have
you.
Thank you for having me.
It's a pleasure, and it was nice to spend a little time up at summer camp, and then prior to that at your hometown there, or at least your current hometown. How did you went to school in California at Berkeley, and you were an engineering student, and then you ended up in the indoor environmental quality world, the bioaerosol world. How did that transition take place?
Well, I can tell you I didn't plan that. I did most of the work prior to my arrival at Colorado was all in wastewater, so water and wastewater, and my training was in classical sanitary engineering, which is a nice way to say sewage treatment processes. So I learned all about the microorganisms in water and wastewater and how to disinfect them and so on, and worked on a lot of treatment plants as a process engineer. And I always wondered how those microbes in that water were getting out and floating around in the air and affecting the operators in the treatment plant. So that's what spurred my interest. But I've been trained in sanitary engineering, and I crossed all those skills over into basically the aerosol matrix and have been working in that for 20 years.
And I'm curious, I after hearing that, I'm wondering, what type of exposure do these guys have that work in the sewage treatment plants?
Well, that's a great question, and it has a very interesting environment, right? We inject lots of air into wastewater to treat it, which is basically the same phenomena as opening up a soda pop can. You got all this air partitioning out of there with lots of volume, very high energy, and it brings microbes into the atmosphere. And some treatment plants are actually completely indoors. So we don't know those exposures, and it's a major aspect of my research program right now. But a lot of the operators at the treatment plants that I had worked in had asked me that, and some were complaining of illness, malaise, so on, and they were associating it with their work. But it's a ripe area of research, and we're working diligently to answer that question.
I have a friend who did that for years, and I always wondered if maybe he'd have more infections, or do you know anything yet with respect to that?
Well, as you know, even with the entire IAQ arena and our building stock, it's a very complicated question to answer. And what we know is moisture and growing things are bad. It's the link between that exposure and human health and mass that is a from a causal sense is difficult to answer, and we don't have that data for occupational exposure in the wastewater environment yet. So it's gonna take a long time to answer that question. And in the meantime, I can say, just based on my experience, most of the wastewater treatment plant operators are pretty healthy people, and we don't see a causal link there, but that question's always there. Is this exposure contributing to any illness, occupational or not, associated with that environment? So, good question, Joe. I'm in the process of answering it, but it's going to be a little while.
Well, and part of the reason I ask is it seems, and Cliff's got a follow up too. It seems that might help us with respect to people who are cleaning up in water-damaged buildings and water-damaged environments and after sewage backflows and things like that. And I'm pretty certain there's not much on their health issues either.
I agree. And it's one thing I'm hoping both industries would do, although they're very different, in terms of their origins, the occupational exposure question is the same, and I'm hoping some industries partners could come together so we can do a survey on that. I think that's a very important thing. And if we surveyed the workers in wastewater treatment and surveyed the workers in remedial cases that are involved with sewage cleanup, I think that would be a very important first step. But it's a big country, and that's a lot of data to gather, but I think it's something very much worth doing.
Cliff?
Yeah, I think one of the common denominators really between remediation and the sewage plant workers is plumbers. And it's, in all honesty, I'm 66 years old. I don't think I've ever seen a plumber wear a respirator. And I think the only time I've seen them wear some sort of protective glove was to protect them against greases or wheels or something like that.
No, yeah, and same on my side of the street. And I agree with you, and that it's a skill, it's rewarding work, but as tough as they are, exposure is exposure, and we really don't know it. I've never seen them use PPE either. They just get in, rip up their hands, and wash them when they're done. But who knows what they're breathing, or who knows what's on their skin? And I think that's worth a look as well.
Well, when you spoke at summer camp, there was a group of you, and it was more of a microbiology of the indoor environment kind of update for the people who attend that. But a lot of your presentation was on research that you had done after flooding events. And I thought that kind of ties into what we're just discussing. Plus, I thought that would be an interesting way to start out today. Can you tell listeners a little more about the types of events you were involved in, and then what type of study you I guess ran after these events?
Boy, that's a
lot.
That's a long-winded story that I'll try to keep tight.
I'm trying to think, how do we break that down in better chunks? But I'm sure you can do it.
Well, Joe's. First of all, I have to say I found that summer school really rewarding to really see, practitioners' perspectives and be able to interact with them on a individual and human scale and listen to the questions and all that. So it was pretty enlightening for me, for someone who's been in the academic sector now 20 years. But to get back to the story I told, I was one of three academics there. James Scott from University of Toronto, Jeff Siegel, who's really a great building scientist, James being a microbiologist, and then me kind of in between, where I'm a civil environmental engineer that's got some updated microbiology research tools. I think it was a nice team, and I think we had a broad spectrum of presentations. Mine in particular was dealing or telling the story of all the floods that I've dealt with since arriving in Colorado, 6 in total. Some in my hometown, others in, unfortunately, Louisiana after Hurricane Katrina, and surveying homes there, La Junta, Colorado, and surveying homes that had been reclaimed from flood damage, all of which had elevated bioaerosol levels. And using the most modern microbiological tools, we were able to show that even after remediation, significant numbers of houses still have either elevated levels of bioaerosols compared to other homes that hadn't been impacted by flood damage, that were of similar age, building characteristics, or so on, or had a different ecology. They didn't necessarily have different loads, but they had different microbes in them. And then floods here in Boulder, in our commercial building stock and in our residential housing stocks, most notably following the 2013 flood that we had here in Boulder. And same kind of story, just coarse differences in aerobiology between flood-impacted homes and not. So that's the theme of the story. Do we have any health data to link with that? No. Do we have we gone back and followed up? No. Just simply because of economy and priorities that we've faced over the last 10 years. So getting money to go back and look at it out over the long term has been pretty difficult, to say the least. But I think we've brought some new tools to the industry, which I'm hoping we'll talk about later in the show. Is that a good synopsis?
It is, and I think we can go from there and maybe pick out some specifics that I think our listeners would be interested in. So maybe we'll start with the foundation. In each of these cases, you were dealing with floodwater, and that would be categorized. We talked about the fact that you are familiar with the IICRC's categorization of water damage. There's either category 1, 2, or 3. And let's start with, I assume in each of those cases, these were considered category 3 black water events.
Yes, and that was simply, so these were freshwater floods, very close to freshwater river sources, very close, with exception to New Orleans, right, which was brackish water. Mm-hmm. That was Pontchartrain and the breaching of the canals and so on. But here in Colorado, they've all been, pretty adjacent freshwater and some would argue pristine freshwater sources. So these were all category 3s based on contact time. So it was several days before en masse we could get to these houses and, get the water out of them, get them dried out, get the materials out and so on. So yeah, unfortunately they were all category 3s.
And when you did the background, before we go there, I've got a text. I want to first get the text question. Speaking of foundations, did the houses that you dealt with have basements, crawl spaces, built on slabs, a variety?
Yeah, at least the houses in Colorado are all, not all, in Boulder, which is, most of it's a relatively young city. They're slab just below grade, and we have a very hierarchical floodplain here. So the houses not in the floodplains have, full built-out basements. Those in the floodplains don't. So most of what we were looking at, and because it was a pretty big flood, were newer homes with basements that had been built out and inhabited. Okay. So in that case, and of course New Orleans, that's not the case, right? There's no basements. The water table's high. They're below mean sea level. They're surrounded by water, so no basements there at all. And in La Junta, Colorado, the same thing. No basements there.
And you used various types of assessment in these buildings, and maybe you could tell listeners a little bit about the differences between, and I'm trying to kind of group them all together for a reason. Hopefully that'll kind of, will help pull this together a little better in the end. But can you tell listeners a little bit about how you assess the conditions going in, and then how you assess them after the remediation? Well,
we use the same tools, and grossly, at least in the most modern terms, what we can do now is we can go in and we can ID who is there, as well as get a direct count on how much is there floating in the air. So the most modern method is to actually take an air sample and pull the DNA out of it and sequence it, and you can tell exactly who's there with a reasonable degree of confidence. And fortunately, there's been all the work that's come out of AIDS and cancer research and high-throughput sequencing has now crossed over into the environmental sector, and those tools are available to us. Is there some uncertainty in it? Yeah, but it's pretty good. It's pretty definitive stuff. So, I can take a surface that's been wetted, or I can take an air sample if I can get enough air, pull the DNA out, sequence it, and have a relatively high degree of confidence of what microbes are in there. So that's who. And then there's how much. And we've used microscopy in the indoor quality sector for a long time, right? Take aerosol cassettes, Burkhardt samplers, and we go in and try to ID not only who's there based on phenotypic, that is, what do the spores look like or the microbes look like and how they stain, but we can get a count. And the microscope doesn't lie. And so we can get, if we take an air sample, we can catch them in an impinger or on a filter and go to the microscope and directly count. And there's much like high-throughput DNA sequencing, there are now high-throughput automated microscopes that can help us with the counting and take the human factor and slowness, take subjectivity and slowness out of that process. So this new generation of optical-based particle counters can give us a total count of particles, OPC, which many of us use, but there's also fluorescence-based instruments that can give us the fraction of the total particles that are bacteria, fungi, pollens, or spores. So we can get a good idea in modern terms of who's there from DNA and a good idea with high-throughput microscopy of how much is there. And we were able to bring these tools to bear both before, during, and after the flooding events in these venues. So the idea is to hopefully after flood and after the remediation, the construction's been done, the techs have been in, that what's present after the flooding is the same or less than what happened before the flood or in homes that haven't been hit, and that the ecology is not threatening. There's no pathogens. It's not markedly different than what's outside and so on. So that's the idea.
And Cliff, go ahead.
Yeah. Mark, what sort of difference, if any, do you think the type of construction made? It would seem to me as a former remediator that, if I was dealing with poured cement or if I was dealing with exposed concrete foundation, that would be relatively easy to remediate compared to, some framing materials that they decided to leave in and, an area that was inhabited, previously. They're trying to save money. They may not have insurance. The insurance company's trying to save money. Was there any difference in how raw the places were following remediation?
Boy, that's a great question, and is a multi-million-dollar question. And as you heard in summer camp from Jeff Siegel and from those who've been in the business of a long time, materials are vastly different. It makes sense that their ability to host microbes when wet is different, based on their porosity, based on their ability to hold water, based on how much organic carbon they would have available if wetted. We heard Hal Levin say, and Jeff Siegel as well, drywall is not drywall, right? They're not all the same. And one wetted can produce certain microbiology while another wetted would produce other. To get to your specific point, about concrete and wood frame and all that, everything that I've been involved with, fortunately or unfortunately, has been wood framed housing, on a simple slab foundation. So I haven't been involved with a breadth of diversity in building materials simply because I'm in more of the modern West, and New Orleans, at least in the areas I was, Gentilly, Ninth Ward, St. Bernard, was all relatively older houses, wood frame houses on simple elevated slab or stilts, just because they're water tables there, and those were the building materials they had. So I don't have a lot of experience with this other stuff, and I think it's a great question and something we need to look at. Big country, lots of different building materials, how they behave when they're wet in those different climates. Boy, what
was interesting is I also did some building investigations post Katrina as well, and one of the things that we found, which was at least unusual to us, is typically these homes would have a flood line. You could see the high water mark. And what we found unusual was generally below that water line, there was very little fungal growth on materials. But yet above that, there was all sorts of fungal growth. And, we weren't sure if it was oil in the water or pesticides or other types of chemicals, or maybe, bacteria had an opportunity to consume food source and, fungi didn't have anything to eat or whatever, but it was kind of unusual, we thought.
Yeah, and it's, that question still looms. And, the water chemistry in the Katrina flood zone was, really varied, clearly, polluted with wastewater. petroleum hydrocarbons. I mean, it was quite a mess. And you bring up a good point. Maybe the building materials that were saturated with this, relatively polluted water had some condition when the waters receded that microbial growth in mass couldn't progress, but where the water had wicked above these lines, and I saw the same thing that you did in many homes, that was these were moist surfaces as opposed to saturated surfaces, and they didn't have the same response at all. And I remember seeing exactly what you're saying, and unfortunately we'll never know. There were a lot of investigations, into the water chemistry there, some done by my colleagues here, but those water samples were grab samples. They weren't composite. They were all over the place. We didn't have them in real time. It was completely after the fact. So unfortunately, we'll never know. And it's a mess we're still mopping up, and it's a continuing story that I hope we have the resources to go back and check out.
You mentioned conditions, and I want to go back a step and then come forward again. We mentioned condition 1, condition 2, condition 3, which is the way the IICRC S500 breaks these things down, primarily based on where the water came from, so the origin of the water, but then it also can change based on time, etc. And I'm just wondering, as a scientist and a researcher, do those conditions make sense to you? Is it something you think we could do a better job with as an industry, or some general thoughts?
Well, I mean, I think as an industry we can all do a better job, but we've got we've got a huge task. We've got a large building stock, limited resources, and it's kind of all over the board. When you get to a job, what resources would be available to do it, what the job looked like before you arrived. So I understand where the IICRC regs came from, and they make sense, right? Was it fresh or dirty water source? How long has that water been in contact with the building materials? So it's kind of, is it white, brown, or black? Category 1, 2, or 3. But the fact of the matter is, when we get in there, it's could be anywhere in between, and 3 sizes doesn't fit all. If you really back up and look at it, it's a continuum. And if we came at it with some of the tools like I was discussing with you earlier to try to get an assessment, was there sewage contamination, even though it has been in contact with the building surfaces, say, for more than 72 hours, that water's still clean. It was a tile floor and that was clean, and the potential to get microbial growth afterwards isn't particularly high. Why would we have to have it a category 3? So I would much rather look at this like a continuum of things with contextual tools that can tell us this has a high probability for growth, this doesn't, regardless of contact time. How dirty was it? How dirty is it? Is there microbes growing in the water now? And this is bar, a sewage spill or something like that. But I'm hoping that we can have a different approach, a continuous approach in the future, rather than these 3 discrete, small, medium, or large, or black, brown, or white, or whatever you want to call it. But I certainly understand the logic of the approach, and it's conservative, right? Mm-hmm. It's conservative. So that's not a bad thing. But we may be spending a lot of more money or time because of that. And I'm hoping the insurers and the remediators and the property owners can get together and look at it that way in the interest of economics rather than being conservative. And again, in the absence of wastewater, right? A sewage spill or fresh water bringing sewage into it, or a sewage line break in a property.
Well, that's always go ahead.
You remember 9/11? After 9/11, we had that the color-coded threat. Threat level 1, 2, imminent, and so on. It was color-coded.
Right.
And they were discrete, right? They were discrete. Mm-hmm. Red meant it's going to happen, we behave a certain way. And really, it's a continuum, right? It's a rainbow. Threats come and go. They wane. It's not, the only thing that I view like that is pregnancy. Either you're pregnant or you're not, right? Yeah. That's not how these things work. That's not how these things work. There's a lot of gray between, or not a gray, but there's a lot of slough between the boundaries. And if we can, the new tools can help us do that. real-time microscopy of water and air, DNA, just counting dirt, right, in the water. Is there a high potential for microbial growth or not? How much organic carbon is there? Maybe a 3 shouldn't be a 3, it should be a 2. Or maybe some clear water's got a bunch of pathogens in it, Legionella or cholera, and you should treat it like a 3. Who knows? But I think it's another ripe area for research.
And some of the new tools may help us with that. I agree. And, one of the things you sort of mentioned is, when that water originally gets into the building from whatever source, if there are not pathogens in it or there are not certain bacteria in it, then they're not ever going to be in it, I guess, or viruses, etc., just because it sits for days. Now, you might get fungal growth or, other things, but is that accurate to say?
Accurate is not the word I think we want to use here, Joe. I think, is it appropriate in context? And you raise a good point, and I was trying to do this by way of example. You flood water on a tile floor that's clean in a hospital or bathroom or whatever, and it sits for a couple of days. It's fresh water on a clean tile. Maybe that's wicked up into a little bit of the drywall, or if the wall's tiled, you're not going to get a lot of penetration. Why should that, after 72 hours, be considered a Cat 3?
Right.
It's right. It doesn't bring a lot of microbes in. There's not a lot for microbes to eat. Likely not going to take off, right? Oh. So that's the context, the appropriateness I was trying to bring in there. Maybe in Pennsylvania they call it accurate. That's cool. But yeah, I would agree with you. I would definitely agree with you.
Okay.
So there's other things ICRC doesn't look at. It's just time and source, right?
Right.
And it's more complicated than that. And maybe we can just add a couple more tools to help us with those complications.
Well, in the second half of this interview, I'd like to talk a little more about some of those tools. So we've kind of set the foundation. We've got to stop and thank our sponsors and pay a few bills. But when we come back, I want to go into a little more about the results you got in the sampling after the flooding, and then move into the new tools and techniques available to maybe be able to make it a little more accurate, for lack of a better term at this point. So let's stop and thank our sponsors. We'll be back with Dr. Mark Hernandez in about 90 seconds. And thanks to our association sponsors, the Indoor Air Quality Association, a nonprofit multidisciplinary organization dedicated to promoting the exchange of indoor environmental information through education and research. Visit them at IAQA.org.
The Restoration and Specialty Cleaners Association, who have been serving the needs of and advocating for their members for over 30 years. Remember, TRISCA is your link to industry training, certification, standards, and events. Their website is trsca.org. Thanks to our advertisers.
Gray Wolf Sensing Solutions, who use advanced sensor technology and embedded computers to provide superior environmental test instrumentation. Visit them at wolfsense.com.
Legends Environmental Insurance Services, the experts in insurance for environmental consultants and contractors for over 20 years. Visit them at legends-enviro.com.
And Particles Plus. They are engineers and manufacturers of feature-rich particle counters, air quality monitoring instrumentation, and vacuum pump technology. ParticlesPlus.com. Count on us.
And, of course, our marquee sponsors. John Donn Products, where restoration and abatement contractors shop. Visit them at their website, jondon.com. That's jondon.com.
CleanFacts, the #1 information source for cleaning and restoration professionals. Check them out at cleanfacts.com.
IAQ.net and Healthy Indoors Magazine, a free online digital magazine for industry professionals and consumers. Subscriptions are available at IAQ.net. Please be sure to thank our sponsors for their support of IAQ Radio when you inquire about their services and products.
Okay, we're back for the second half of our interview. We've got Dr. Mark Hernandez from the University of Colorado. And Mark, I'm curious, you did a lot of sampling before and after remediation of these flooded buildings, and I'm wondering, I know this can be difficult to do, if you can kind of summarize for us your thoughts on how effective remediation was and what kind of results you were seeing before, during, and after.
Boy, that's a biggie. I'll try to do that in words. And I'm going to go back to the analogy that I used earlier, not the analogy, but the categories I used earlier in the show, and that is who's there and how many, right? Both of which are important to us. And the unfortunate thing with floods is you rarely know when they're going to happen, how large they're going to be. If you do have warning, you might not be able to get your sampling equipment in there. So getting in before is a rare event. However, we did have that opportunity in at least one commercial office structure. And there's a timeline to this too, Joe. So some of the stuff we were doing 20 years ago, right, in the late 90s, and at that time, all we had was culturing and microscopy. We didn't have DNA stuff. We didn't have real-time particle analysis and those types of things. So we had to rely on the good old tools, and these were, impingers, Anderson impactors, and so on. But even with that information, we found, at least in the small numbers of homes that we had observed in La Junta, Colorado, that both the numbers and who we could grow, we were using culturing at the time, were different. Several months after the remediation had been done, and they had been re-inhabited. So as judged by the classical industrial hygiene tools, we did see differences both in number and what we could grow. Fast forward in 2005 in Katrina, 2008, 2013, 2014, and 15 in Colorado, and using these new high-end tools, we saw the same thing. So early on, and when DNA sequencing was becoming available to environmental science, we found differences, at least between outdoors and indoors, and buildings of similar construction that had not been flooded. And then as this DNA technology has matured, that answer is still stayed the same. Months after remediation, after occupations occurred, we still see differences as judged by modern DNA sequencing. And it's got its own slop and variance, but within what variance we anticipate with it, different microbes present in some microbe families. And then when we have used this real-time microscopy, we've also seen differences basically in load. Particle load, both biological and not. So hopefully that wasn't too much of a mouthful. Short answer, who's there and how many are there have been different, even with modern remediation, over the 20 years and the 6 floods that I've been involved with.
And when you say different, is that, do you see less? Do you see...
Well, it's the same or more in terms of microbe numbers between indoors and out, or homes of similar construction. And then different is who on a very coarse level, microbial families, we call them. And in some cases, genus and down to species level, if you can get into the really expensive sequencing, which I'm hoping is going to get cheaper. But yeah, it's this, in terms of how much, it's been the same levels, not statistically different, or more. As much as we can control. And keep in mind, these are relatively small studies, right? We're talking about tens to 20, 30, 40, 50 homes max, and 1 or 2 commercial buildings. So this is not an exhaustive study by any means. Sure. Whereas, we see with, the storm damage in recent memory, Sandy through a lot of the superstorms that we've seen in the Southeast, and we're talking about thousands, tens of thousands of homes or buildings, not just homes. So we have a little snapshot into really what's a movie in very different building stocks, and getting that data would be an important thing. And I'm all for now monitoring in high incident areas that haven't had floods, right? So we can get get a baseline in very different normal building stocks before anything ever occurs, and we have a better idea what normal is, because right now we don't.
Cliff, I think you may have a comment.
I do. Thanks, Joe. It goes back to, a couple of things Mark said, who and how many. I think in terms of the remediation, I think it depends who was there, Joe, and it also depends how many claims they had. If you have a restoration company that, very competent, and they send their best crew out there and they do a high-level remediation, I think it may affect the results, versus a situation where the company has too many claims, that they have a bunch of temporary people, their staff is stretched, the staff is overworked, and maybe they're taking shortcuts. But I think the quality of the remediation, and we've talked about it many times, it's really not trying to kill stuff. It's really about deep cleaning, and deep cleaning takes time and it takes effort, and I think that would make a significant difference.
Yeah, I'd have to agree with you on that, and I've seen it. I've seen what I consider some of the best of the industry. I live in a relatively wealthy city. When that flood hit here in Boulder, there were lots of insurance. Some of the bigger remediation companies were here, lickety split. That happened at my university and in my commercial building when we had a flood there, and these guys were in. It was a full court press. It was just impressive. Incredibly well organized, state-of-the-art equipment, not leaving a stone unturned, and at least by modern methods, and my university building being an example, without naming the company that did it, I couldn't measure differences with real-time microscopy. I couldn't measure differences with modern DNA techniques. These guys were organized on it, multilingual. They had multicultural crews, age and gender stratified. It was really something. And within a month, we had our building back. It was pretty cool.
I want to move on to another topic here altogether, although they're kind of, interrelated, because you're talking about some of these newer methods and so on. And recently you hosted and helped organize the Microbiology of the Built Environment Conference. This would be, I guess, the fifth one, all sponsored by the Sloan Foundation. And I'm wondering, we did a show from there with several of the speakers, and I thought it was well received. And then I'm just wondering, from your perspective, what were the big takeaways from this 5 years of research and spending a lot of money? What new things did we learn that and let's I'm sure we learned a lot of new things, but let's kind of move it toward research to practice. What did we learn that might help us inform practice?
Boy, that's a great question, and I was fortunate enough to be the organizer of this, and together with the project officer from the Alpha P. Sloan Foundation, we were able to craft this program. Early on, a lot of the investment in around 2010 was going into what I'm going to generically call bioinformatics, and that is the ability to take a sample, surface, water, aerosol, doesn't matter, and in a short period of time, using genetic methods and computational methods, get an idea of who's there with a relatively high degree of confidence. So that's where a lot of the investment from the Sloan program went in the early years. And over time, over the 5 years, it really transitioned from the investment in being able to confidently inventory the microbiology of inside to what's the effect or the potential effect of some of the things we've talked about today, building operation, building materials, on that microbiology. So I really got to see it transition from dominance of biology and focus on the methods to dominance of using those methods to survey microbial response to what is there, i.e. building materials, wetted or not, and how the building is maintained and operated. So there was quite a transition, and granted that it was dominated, as you saw, Joe, by those of us in academics. I think that transition was useful, going from microbiology, crossing the chasm to engineering, and that's still in its young state. And when I got to the summer camp, I was humbled by how the practicing field without these tools has progressed. And you know, we've got these expensive ways to do assessment, and the practitioners didn't, but clearly what they're doing they know is beneficial. Clearly what they're doing is getting rid of moisture, you know, in mass, you know, on a large scale. And did academics know that water's bad? Yeah. Water where isn't it should be. So I think we're getting close to a transitional point, to answer your question, with that programmatic investment from Sloan, where these could be available if they get cheap enough and if they get standardized for the practicing community. And I think that was really the benefit of Sloan's investment. You know, the last of these conferences will actually be held in Washington next fall, and it will include agency representatives to see if funding in this arena for both fundamental and transitional research can continue.
And you know that the combination, you know, bringing the researchers and the practitioners together was interesting, and kind of going along the lines of what you just mentioned. I was able to talk briefly to James Scott while at summer camp, and he had mentioned on the show, I'm going to say 6 or 8 years ago now, let's say 5 years or more ago, we had him on. He talked about the past, present, and future of microbiology. And at the time he was saying, you know, within 5 years, even less, we'll be able to take these samples, get all this information, it'll be inexpensive, and that's all happened. But then, you know, when I talked to him a little bit more about it, I said, you know, we can get that information, we can do this type of sampling, but the problem, according to him, is we just don't know what to do with all that information. We get a lot of it, but we don't necessarily know what it all means. Does that seem, does that, you know, kind of go along with what you see? Yes,
and I think what James is saying, I mean, and he's a very smart, visionary guy, you know, and James wears both hats, right? He runs a very good academic program in U of Toronto, and he also founded and runs a analytical laboratories to support indoor air quality research, right? So he gets both worlds, and what he's saying is we have so much information now from the genetic sequencing, or the potential to get so much information, that there's a lot of noise, and we have to figure out what signal and noise. And that's in the absence of pathogens, right? We don't have Legionella, we don't have tuberculosis, we don't have Stachy, and so on. So the DNA stuff is really great for identifying the bad guys, but it's turning out to be very rare that the bad guys are showing up, at least with genetic sequencing. But it is a mountain of information, and discerning what's noise and what's signal is kind of tough, you know. And to give you an example, when we first started this business, we'd get maybe 1,000 sequences from air samples in flood-damaged homes. Now we're getting millions. So identifying what the dominant players are, identifying the uncertainty in that signal is kind of the next challenge for this bioinformatics community in the context of exposure in the built environment, like you just said. So I think that's what James is referring to, and I would agree with him.
Yeah, because at this point we're still seeing a lot of spore trap sampling, and we have better methods, but what do you do with all that information, and how do you interpret it, and how do you put it into action? I'm hoping that's where this whole microbiology of the built environment is heading, as opposed to, you know, just learning more about the microbiology. Let's figure out what to do with that information once we have it.
No, agreed. And that's, I think there are in the Sloan cohort and at least the students I train, I think that Jeff trains, that some of the people that were at the summer school, our trainees, we're sending them out with that purpose, to use this in a translational but cautious way. These are not silver bullets, but they could be useful tools. And that's kind of the idea, is from our labs train kind of these next generation of industrial hygienists to transition these tools in a thoughtful and useful way. But it's going to take time. We'll like we have in the medical realm, it's going to converge to some useful tools. At least that's my hope.
Well, and maybe there's some middle level here. We've got the old techniques that have been around for many years, and we're looking at kind of the future with this next generation sequencing stuff and all the DNA and so on and so forth. We had you on a show not long ago discussing, I'm kind of looking at it as a middle ground, and the work you did for the folks at Instascope, with, looking at particles and the size and shape of particles and then how they fluoresce and so on. And to me, that's kind of a middle ground. It's more useful information, at least. It's kind of at a level where we can actually take that information and make some decisions based on what we see. The first question I had for you is, are you involved, financially with the Instascope other than doing some of the research for them?
We and I'm going to speak for all of us in North American universities, the engineers and researchers, is we cannot have any interest or equity positions in the research sphere, and that is by contract, pretty much by and large, in academia. So to get to that kind of question, that is the ethical standard in our industry. Is we can do research and we publish our results with emerging technology, or we do R&D, but this is all blind, peer-reviewed stuff, and we can't have financial interest in doing any of that. I do have colleagues that have left academia to go work for companies when they see promising R&D come out, or they've been, part and parcel of the industrial or intellectual property. But no, we can't
Okay.
unfortunately do that, and that's that's it. No stock options, but research is fun, and we do this job because we love it, and like to see this technology forward in the profession. But I agree with you. I think that there's a lot of middle ground to be had. We gotta side by side use these new techniques with the classical techniques to help the industrial hygiene field be able to interpret this new data, and we got to standardize it, Joe. There are not standards in DNA sequencing. We get a sequence, and how we arrive at those sequences, and what software we use, and how many samples we take, and all this feeds into the uncertainty and the signal-to-noise ratio. So we have to, there's a bit of work of standardizing before we cross that hard chasm in the industry, bar looking for pathogens. That's just FBI stuff, right? It's how we bust criminals. It's how we bust pathogens. But when you do these big ecological studies, we got to have a better handle on uncertainty.
I'm wondering if maybe the one of the attempts at doing that is how the ERMI program developed. They were able to look at the DNA of microorganisms, and then they grouped them into those that were commonly found in outdoor samples and those that were commonly found in water-damaged buildings, and they put together the relative moldiness index. Do you see that as maybe a potential model for how you could take some of this next generation stuff and make it more applicable to the real world?
I do. ERMI's controversial, but the framework of ERMI is not. I think it's a brilliant approach. The architecture of that, where you get a distribution, right? You essentially get a probability distribution, or what I call a rank order distribution. It is it's a brilliant approach if you've got enough data. And yeah, that's where I do think I see it. I'm not going to use ERMI or that acronym, but the idea of using distributions, is your home in the 50th percentile, 25th percentile, or are you an outlier at 90 percentile in terms of microbial load or certain types of microbes, and I liken it to those of us who have children. You bring your kids in, and where am I on the growth chart? Is my child undernourished? Why are they not growing? Why are they not heavy enough? Or are they an outlier on the other spectrum? And what I don't want to own is a property with, I'm 95th percentile, I'm off the charts in terms of microbial load or a certain type of ecology. And if we had that information broken down that way in the framework of rank order, I think it would be really useful for practitioners, both in terms of who's there and how many are there. So, and pre and post remediation, right? Are we as clean as other homes in my zip code after my house got wet? I want to know that. I want to know what's floating on the air and the surfaces in terms of the other houses that are similar age and construction near me that maybe didn't get hit by water or have some other unfortunate event.
Sure. And it seems to me that in some ways that's what the folks with the Instascope are trying to do, and you help them with getting the signal, I guess it would be, from different types of microorganisms so that they could at least group it into the fungi and the bacteria and the pollen, etc., and then that you could get some. Yeah. So go ahead.
So grossly, what we did in our research group is evaluate the technology, not the product. And this technology has actually been around for quite a while. It's matured and crossed from the military sector in terms of looking for biological warfare agents to the civilian sector, and it does what you just suggested. It can successfully segregate bacteria, fungi, pollen, and bacterial and fungal spores from each other in a very gross way. It might even be able to, and we're working on this with just fluorescence microscopy in general, separate different families of fungi and potentially pollens. I don't think we can with bacteria. But anyway, that's the idea, is to, get a confident breakdown of the total particle numbers, classical OPC with lasers, and the biological fraction thereof accounting for potential interferences. So I do think that technology is promising, particularly for healthcare settings and ultra clean places, and I'm hoping, and it's gonna work in really dirty places too, which I think the data that's coming out, I think they must have more than 3,000 observations, will help us get to that in a rank ordered way. Research has to come, Joe, but you're right, it's maturing.
And just to give listeners a better idea of some of the work you did there, you actually grew cultures, I guess, of known fungi like Aspergillus fumigatus or whatever, and then you have that bioaerosol chamber, and I'm assuming you're able to release these into the chamber and then look at the signal that came back once you did whatever magic that goes on in that box, and then you were able to tell if that was.
You got it exactly right. It's just a big generic room, 10 cubic meters. I have a full suite microbiology lab. I can grow any bug you want, and we did, including pathogens, MRSA, cousins of tuberculosis, Legionella, and so on. We aerosolized those in the room in a very safe and contained way, and then along with fungi and pollens, we used this fluorescence technology to see if we could tell them apart, and using size, fluorescence, and basically those two combinations, we could not easily, but using an algorithm, tell bacteria from fungi from pollen and their fragments. So it was pretty cool. But yeah, you got the plan. We've got a freezer full of any microbe you want. We grow it up into a teenage state, an adolescent state. We aerosolize it at a defined humidity and temperature condition, let it equilibrate with those conditions, and then we take our samples and see where they land. So this thing can tell a blue-eyed microbe from a brown-eyed microbe, a blonde microbe from a brunette microbe, and a fat microbe from a skinny one. You put all that information together, and it helps you segregate bacteria, fungi, and pollens.
But it's not going to give me a specific breakdown of Aspergillus fumigatus versus, Cladosporium, whatever. Is that accurate to say?
It is not. It can't discriminate between genus and species, at least not yet. Some microbes are so profoundly different, Stachy being one of them, that potentially in the future, if we've done enough cataloging, we might be able to single out a certain genera. And we've seen that with Stachy. We've seen it with Bacillus statospores, certain biological warfare agents. But the jury's still out on that, and I would be cautious with that. But right now, for acute ecological, biological loading, what's floating around in air, I think it's a really nice counting tool.
And you mentioned when we talked, and I realize we're running real low on time, but there's just so many things I want to talk to you about. You mentioned before we started the show that you had just published an article, and was an atmospheric measurement, I think, and could you tell listeners a little bit more about that and just kind of an overview of it and where they could get a copy of it if possible? Yeah.
So it's a relatively, it's a very established journal, Atmospheric Chemistry and Physics, and it has a measurement techniques arm, and it's called just that. It's called Atmospheric Measurement Techniques. And what's the beauty about this is it's an open access journal. It's highly respected, and you can get it free. So you can just go online and Google and look up Hernandez bioaerosol fluorescent catalogs, and it'll pop right up, and you can download a PDF. This next generation of academic journals is really nice because it's free and open to the public. But in a nutshell, you'll find in there what we just discussed: a scientific report that unequivocally shows that with fluorescence-based microscopy, you can separate bacteria, fungi, their spores and fragments from pollens, and that the colored signatures that these new generation of high-throughput microscopes can distinguish are really very distinct. So it uses old-school optical particle counting technology, right? Just a laser and use light scattering to size them. But together with fluorescence information, what this particular investigation shows is with a large number of fungi, bacteria, and spores, there are more than 50 in this investigation, that you can really separate them with this high-throughput microscopy. So it was pretty cool. It was, from a research or a translational research standpoint, it was really fun to be able to work with this emerging technology to show that for an applied context. But Atmospheric Measurement Techniques, it's called, is the journal, and catalogs of fluorescence and optical properties are the keywords on there, and it'll pop up, and you can check it out. You can read over a cup of coffee in 10 minutes. It's pretty available.
You just mentioned something that really caught my attention. I don't know why I hadn't picked up on this before, but you said it would pick up both spores and fragments. Did I hear that right? Yeah. Okay, because that has been as long as the fragments are large enough. Okay. I'm not talking about tiny stuff. So, roughly the cutoff is about 1 micron in diameter. That's the size of a dehydrated bacteria floating around. They can go down to about a half a micron, but then resolution starts to get a little dodgy. Okay. So we know we can see pollen in their fragments. We know we can see fungi in some of the larger fungal fragments if the spore split. Bacteria, no, that is not the case. But we can see whole cell bacteria and distinguish them from fungi and pollens. Interesting. And I want to say one more thing about that report, and it was really rewarding. We worked with NOAA, the National Oceanic and Atmospheric Administration, in addition to the senior scientists at Droplet Measurement Technologies. So it was a nice industry agency, private, a university cooperative that did this, that came together to make that investigation happen, and that was really really nice. Daryl Baumgartner from Droplet Measurement Technology and Ann Paring at NOAA. Each of us had overlapping expertise, and it was just
great collaboration. I think we came out with some good information for the industry.
What was NOAA's reason for their interest in that?
Oh, this is very interesting and relevant to climate change. So there's some information coming out in the scientific community that suggests that airborne microbes can serve as nucleation for raindrops and precipitation, certain types of microbes. And what NOAA is interested in is using this device for large-scale environmental studies, and they're flying it on airplanes. I was part of an investigation that went to a volcanic island out in the middle of the Indian Ocean and used this technology, used a WIBS, a fluorescence microscope real time, and they're looking for microbes in the troposphere and looking for interactions with clouds such that they believe, or the hypothesis is, and this is what NOAA is in the process of researching, that microbes participate in precipitation. It's pretty interesting, especially as the atmosphere becomes more humid. This idea that microbes participate in precipitation events, it is very ripe for research and very important for the planet. So that's why NOAA is involved.
So maybe it might help in like cloud seeding and things of that nature down the road?
Yes. Yep. Or understanding how biology plays into environmental processes. As the planet warms, the hypothesis is that we're going to see more microbes in the atmosphere, and that may play a role in precipitation. Now, keep in mind, this is all burgeoning scientific theory. This is coming out and has not been proved out yet. But we're in the process of, and this technology is a part of it, of using or of vetting that theory. It's hard to do, right?
Yeah, fascinating.
and expensive. Flying this stuff on airplanes and climbing to the top of volcanoes with expensive scientific equipment is not the easiest job. So I salute NOAA for doing that. And anyway, I feel grateful I was able to participate in that project.
Yeah, I'm glad we were able to have you on and enlighten listeners as to other ways that this type of research is being used in areas maybe a little outside of the built environment, but still very interesting. Cliff, before we ask our final question, is there anything you'd like to follow up on, or any questions that you have?
No, Joe, I'm good, thanks.
All right. Well, Mark, it's been great having you. Before we let you go, though, we always like to give you the final word. Is there anything we missed, or anything you'd like to add that we missed, or that you'd just like to add in general before we wrap up for the day?
No, just thanks for having me, and I'm feeling real fortunate. It's such an exciting time, with this new tech, and to be part of the potential crossover of this stuff into industry. And I love the summer camp and learned a lot from the practitioners there. So that's it, really.
I hope you can come again because it's a great learning experience, and I think you have a lot to offer for the folks up there that are hungry. That's a hungry group for knowledge up there.
That's just a hungry group.
That too. Well, again, thanks for joining us.
Joe took care of everybody, that's for sure.
Yes, he did. So. We're going to try and get him back. He hasn't been on in a long time, and our 10-year anniversary is coming up in a couple of weeks, so I think he'd be a great guy to bring back on. So I'm kind of throwing out a little seed right now and hoping it germinates down the road. But thanks again for joining us, and I look forward to seeing you again somewhere down the road. Hopefully next year I'll learn in advance when that next Sloan thing is in D.C., because that's right down the road from me, and I'd love to see where that stuff's all going in the future.
Very good.
All right. Well,
thanks, gentlemen.
Thank you, Dr. Mark Hernandez, for joining us this week on IAQ Radio. This is Radio Joe Hughes saying thanks again to our guest, of course, to my co-host, the Z-Man, Cliff Zlotnick. Cliff will have a very interesting blog, I'm sure, this week. At the controls, John, you got to have faith. Most importantly, our growing group of loyal listeners. Come back next Friday at noon for the next live broadcast of IAQ Radio.
This has been another IAQ Radio production.